Welcome to Chapter 12, where our topic is developmental genetics. It is complex, but it is fascinating because it's how we are able to form from a single fertilized egg. And here is a fly embryo, and it's been you're looking at is all sorts of different gene expression in these early fly embryos. You're detecting different proteins that are being synthesized using antibodies that have fluorescent dye on it, and you can see their concentration differs in each of these different units. So it looks like it's a single cell, but it's actually multiple cells, but they're expressing proteins at different levels and in different locations all at the same time here. And so we'll be talking about spatial and temporal expression of genes. So we're back to the central dogma yet again. All right, developmental genetics is defined as the study of how the instructions encoded in genes control and coordinate development of an organism, beginning with fertilization and, of course, ending with death. What you're looking at over here is a cleft palate. Anyway, mutations in genes can and do disrupt normal development. Birth defects, in fact, are very common. 2 to 3% of all live births, they have some form of birth defect. Most of them, we don't know the cause. It's estimated a substantial proportion are due to defects in genes that control normal development. Well, that only makes sense if you think about it. So, basic concepts of development are as follows. First of all, definition. Animal development, we are animals, is the process by which a fertilized ovum becomes a mature organism capable of reproduction. Now, a single fertilized egg, in order for that to happen, must undergo division. So we go from one cell to billions of cells. And then differentiation. We have to have all those different cell types, red blood cells to carry oxygen, endothelial cells, epithelial cells, all these different types of cells to get the tissues and organs in order to have a fully functioning individual. This involves a species-specific body plan. The arrangement and pattern of different parts of a body is going to be specific for a species. So here we have a developing human in utero, and here we have a corgi puppy. And why would I put a corgi puppy there? Because I am a corgi puppy lover, and I have 3 corgis at home, which is why I do all my recording at work, because otherwise you'd hear lots of barking in the background, and it wouldn't be particularly professional either. Now, each nucleated cell of an individual has the exact same genes, what you inherit from your mom and dad. If that cell has a nucleus, all of the genes are in there in every single cell. So, the question becomes, how do cells with the same genetic information form different cell types? And then how do they come together to form an organ? And then how is the body plan determined that we have the feet on the legs and the hands on the arms, etc.? So again, we have a cute little toddler here, and again, a little corgi puppy trying to climb the stairs. So, in order to study this, of course, we have to use animal models. Obviously, ethical reasons are pretty high on this. And what we have found, though, by studying this, is that genes that we found in animals are conserved if we're talking about, mammals and this sort of thing. And so we can study these pathways in one animal and apply it, of course, to another. And we see that the same regulatory signals and pathways are used more than once during a development. They'll be turned on, then they'll be shut off. And this happens again and again and again. Now look at one of the ones that's been really helpful for us for studying is, believe it or not, a worm. A roundworm, C. elegans, is a particularly useful eukaryotic microorganism. It's easy to breed and maintain, but the tissues cannot be cultured. And then, as you can see, some of the others, we use frogs. Zebrafish is really big right now. And I used to use a lot of mice and rats. You don't feel quite so bad about killing mice and rats, although some of the inbred strains are pretty cute. And then there's what we call ectopic expression. And this is, of course, the expression of a gene product, a protein, okay, in an abnormal location. So we have things like PAX6 mice or PAX6 humans. And the same sort of thing would be in a fruit fly, eyeless. These are all genes. When they are italicized, as you see right here, that means we're talking about the gene. When they are no longer italicized but straight up, then we're talking about the protein. So I think I said that probably early on, but I think it's best to recall that. Anyway, these are all homologous. In other words, we all have these same genes. They're called slightly different. And what are they encoding? They encode transcription factors, which are important, eyeless, and eye development. And so you see here some of the evolutionary relationships among the homologs. That in this case, we don't have an iris and iridia. And here with the mouse, we have small eyes. And here with the fruit fly, as it says, the misexpression of eyeless and tissue failure to become an antenna causes formation of a normal but misplaced eye. So there are some major processes in embryonic development. I know your textbook, again, doesn't go into this, but I feel that it's worthwhile to review. So it'll make understanding the cases in this particular chapter a little bit more informative. And again, with your rewrites of your discussion, particularly the summary paragraph. I didn't see a lot of you pulling together the different genes because sometimes they had some overlapping functions. Some of the groups did pretty well on this. Others just restated what the gene would do and not think about how they might complement or synergize or be antagonistic. So the major process in embryonic development, axis specification. You've got to define what's ventral, what's dorsal, what's anterior, posterior, medial, lateral, left, and right. Then you got to have the right pattern formation. This is the steps in which the cells are arranged, and they got to be the right cells in the right tissues and organs. And then you've got to form, of course, the organs and the limbs. So that's organogenesis. Now, in order for this to occur, we have to have interaction of the differentiated cells. And this interaction is mediated by what we call induction, where the cells of one embryonic region affect the cells of in another, and polarity. And this, of course, is going to specify direction. Now, here's are some examples where direction and things get messed up, and organ formation doesn't completely, there's mistakes in the organ formation. Of course, we see the thalidomide babies that are classic here, but you can see where we're not getting both legs and this baby in the middle, and some very upsetting pictures here of things that go wrong during development. In fact, if you think about it, it's amazing that it works most of the time. So let's start our case. Part 1 and 2 involve obviously a married couple, Jane and Albert. And they have an ultrasound at 18 weeks, and they find out that their baby is small and has a horseshoe kidney and an atrioventricular canal defect. So they do go ahead with an amniocentesis. Karyotyping reveals it's a normal number of chromosomes, and it's a girl. And they test for a common deletion. Well, I shouldn't say it's a common deletion, but they test for a deletion that is associated with the symptoms that they're seeing in the child at 18 weeks. They also do a fetal echocardiogram, so they see a large heart defect that will require surgical repair after birth. And at birth, they also see that the baby has unusual features and goes into respiratory distress, requiring intubation. So what's going on? Well, the newborn has what we refer to as a malformation syndrome, and this is affecting multiple organ systems. Now, if the pattern can be recognized, this really helps diagnosis a great deal. Now, there are really three major forms of anomalies, or at least we classify them into three. So every time I think about it here at Concordia, we always have Trinity. But anyway, the malformations occur when the development of the tissue is abnormal. So the development, the process has been interfered with. Deformations occur with something outside the tissue presses and distorts the tissue. Okay? And then disruptions occur when a normal tissue, the tissue's normal, but it gets damaged. So you can see that these are really three different processes, and so they warrant having different categories. So, what are the patterns of malformations? We can have a syndrome. This is where a set of congenital defects are due to a defined genetic cause. So, a syndrome, we know what's doing it. It's a particular cause or a particular genetic defect or exposure to teratogenic agents such as thalidomide. A sequence, however, notice how we got another trinity here, arises from a primary event that induces secondary effects. An association where we have sets of congenital anomalies whose causes are unknown. So we can't really call it a syndrome because a syndrome we know the cause and effect. And the sequence we usually know, the primary event. So this leads us to our field that we call dysmorphology. It's part science and part art because the dysmorphologist has to recognize both major and subtle anomalies, recognize their patterns, diagnose correctly, and then provide anticipatory guidance. What's going to happen next for the patients, and then the parents, and then what's the recurrence risk. So here's an example of this little girl who was born with the disorder that we have in our particular case in this chapter. And here's a little boy with the same syndrome. And as you can see, the effects on the facial features can be quite different in some cases. I think both of them are adorable, but I just love the expression on that little boy's face. While baby Lisa is taken to neonatal ICU to help with breathing and feeding, they also managed to get some CT results. And they find that she has choanal atresia, which they think is accounting for her problems breathing. These are small nasal passages. She has abnormal temporal and inner ear bones, not binds. I will fix that again. And bilateral columbata of the optic nerves, which turns out to be these spots. And then also additional hypoplasia. So all these additional findings correlate with a condition called Charge syndrome. And it's called Charge because you have the columboma. Not quite sure where the emphasis is on this. But these are simply notches or gaps in one of the several parts of the eye, the heart defects, the choanal atresia, retarded growth and development, genital hypoplasia, and ear anomalies and deafness. And this is just showing you the coloboma, whatever. And there's a little notch in here if you look really closely. So, the parents are told that Lisa has Charge syndrome, and they meet with the geneticist. And the geneticist explains that Charge syndrome has a very variable pattern of expression. And not all of the things that stand for Charge are seen as in every single patient. However, feeding problems seem to be common, and she's just going to be small for her age. She might be at recurrent risk for upper respiratory infections, in part because of the small nasal passages. And obviously, with the colobomas, colobomata, the visual problems and hearing loss. I always had problems with pronunciation when I was younger. I think I still do sometimes. Anyway, there's no family history of genetic defects in either the parents' background, but Jane did have an earlier miscarriage, and Lisa is their first child. However, they are told that usually this is sporadic, But they recommend both parents being tested because sometimes there is a rare familial inheritance reported. So they take blood tests to confirm Charge in Lisa, who is beginning to do well. So we'll talk a little bit more about Charge syndrome. The gene affected, this it seems somewhat familiar from my little discussion. Anyway, is a CHD7. It codes for what we call a chromodomain helicase DNA binding protein. So CHD, chromodomain helicase DNA. So what it's going to do is that domain refers to the fact that it will bind to DNA and cause it to change structure in a way. So this is part of 7 is obviously part of a protein family. Chromodomain helicase DNA binding proteins, this is number 7. And it remodels the chromatin, which will then affect gene expression, but they do it during early human development. Expression pattern, think tissue location, central nervous system, inner ear, and neural crest derivatives. In other words, anything that arises from the neural crest. And so this is where we're going to see the problems with the developing child. It turns out that these particular proteins control gene expression by changing the chromatin. They don't alter the DNA, sequences. They do not alter the nucleotide sequences in DNA, but they inhibit or promote transcription of genes in mesenchymal cells. And these are derived from the cephalic neural crest, and so this is why anything that arises from the neural crest, the particular types of things that arise from the neural crest will be affected in this disorder. So if they can't control gene expression in their normal manner, of course, things are going to be expressed at the wrong time and perhaps in the wrong place, and things don't develop properly. What we find most of the time is a deletion. And again, it's on chromosome 8, and it's on the long arm at band 12.2. It turns out a lot of times, if it's not the deletion, it's a balanced translocation between chromosomes 6 and 8. But you can also have some smaller point mutations. And these will lead to, again, premature termination, or what we call nonsense codons. And so this is the sort of thing I'm looking for. If you just give me in terms of mutation, it's a homozygous or compound heterozygous, you're just telling me it's a recessive disorder, because they both have to have, both of the genes, copies of the genes have to be affected. What I need to know is the mutation, okay? And it is autosomal dominant. It can be so mild that the parents are unaware of the presence of a genetic disorder, or it's a sporadic event. So parents are going to need some counseling. Blood test confirms Charge syndrome for Lisa. Parents are normal. They have a 2% recurrence risk because germline mosaicism has reported, even though they're normal. So you've got to explain syndrome, inheritance, and molecular genetics to the parents. And if they don't have a background in nursing or any form of health science or science in general, they are pretty lost. So, and they're traumatized. They have a sick child, and they're having to make life and death decisions. So counseling is so important at this early point. But I know you nurses know this very well. So you've got to connect the parents to other families with similarly affected children so they know what their life is going to be like. And then they're lucky Lisa goes home after a month. Now, let's think about the genes that are required for normal development again. And this is my little picture of them and everything that I've done on PowerPoint. Signaling molecules and their receptors. So here's the receptor on the surface of a cell, but remember, receptors can be inside the cell if the signal can cross the cytoplasmic membrane. And the signal, of course, is going to bind to the receptor. Then we have DNA transcription factors. They're going to alter gene expression. Remember what gene expression is? The transcription of a gene into messenger RNA and the translation of it into a protein, the central dogma. Transport systems to take things in and out of the cell, okay? is something else. ECM is extracellular matrix components. Cells need to be able to bind to things on there so they go to the right place to form a limb. Enzymes that catalyze a lot of these reactions. And again, transport systems. Now I'm pointing to transfer systems in and out of a cell, but transport systems to get the right cells to the right part in a developing embryo is another type of transport system. So all of these things, and there's thousands of genes involved in this, have to be expressed in the proper spatial, the right location, and at the right time. You mix up those combinations and you see some of the things that we saw in that previous slide. So these are common causes of birth defects in these particular genes. Now I'm going to again take a little time to cover some of this which is not covered in the textbook. Just talking about paracrine signaling molecules. Paracrine signaling molecules are secreted from one cell, and this signal is only supposed to impact adjacent cells, okay, so in neighboring cells. So the proteins don't travel very far. It's a very short distance. Now, there are 4 major families of this, and I'm not going to expect you to know these. I just want you to see that these are things that are obviously involved at other stages in a person's life as well. And they have these weird names. Fibroblast growth factor, that seems okay. But then you have things like hedgehog and wingless. And the reason for this is because they were first identified in fruit flies, and they gave fruit flies these different phenotypes. They didn't have a wing, or instead of having a smooth coat, they had bristles, so they called them hedgehogs. And then we have transforming growth factor beta, which is another big family. But each of these are fairly big families. And again, you don't have to learn these. I just want to give you a taste of this because it really is fascinating stuff. And each of them bind to one or more receptors to generate a response. Okay? Mutations in the genes lead then to abnormal communication between the cells. So the cells don't know what to do at the right time and at the right place. So, again, abnormal communication between cells. Let's take a very brief look at disorders due to mutations in fibroblast growth factor receptors. Now the receptors are glycoproteins, so they just have sugars attached to them. They all have a similar structure, and they bind to at least 22 different fibroblast growth factors. Those are the signaling molecules. They have domains, the exterior of the cell, they have what they call immunoglobulin Ig-like domains. They just have a particular shape. They have a transmembrane region, but more importantly, on the inside, all of these are different domains, what we call intracellular tyrosine kinase. And that means that the intracellular portion functions as an enzyme that attaches phosphates, if I could talk, onto other molecules, initiating signal transduction within the cell. Now, fibroblast growth factors are very important in development. They help mediate cell migration, so helping the cells get to where they're supposed to, growth, divide, and then differentiate, go into these different types of tissues. Fibroblast growth factor receptors are widely expressed then, but particularly in developing bone, again, tissue expression. And when they're mutated, they cause many autosomal dominant disorders of bone growth. Not so surprising. So these are just kind of a picture to show you the structure and function of fibroblast growth factor receptors. And all these little bumps here are referring to the immunoglobulin domains 1, 2, and 3. Here's the TM transmembrane domain, and then these tyrosine kinases 1 and 2, and this is fibroblast growth factor 1. If this gets mutated, there's a disorder known as APERT. These are the disorders. Some of these are unclassified. But these are fibroblast growth factor 1, 2, and 3. And then when you look down here, you see all the different proteins that are involved in getting the signal transduction going. Just want you to get a sense of what these proteins kind of look like. And all of these domains are encoded in the different exons in the gene in the DNA. So let's look at fibroblast growth factor receptor 3. A missense mutation in the transmembrane domain, and you get achondroplasia, otherwise known as dwarfism, but we don't like to use that anymore. And then the overactivation mutations inhibit growth of chondrocytes, producing skeletal defects. So they get things like skeletal dysplasias. So sometimes you can get overexpression of some of these, and that causes a defect as well. Less activation, hypochondroplasia. Marked activation, and you get something called thanatophoric dysplasia. And you can see it's much more severe. And this is again showing you the extent of the different effects that you can get from here. And what you're seeing on top here is the domains that are affected. So I just wanted to give you a sense of how this, and so here is the protein, but these are, all these different little areas are referring to different exons in the DNA. And I think that helps you get a feel for how the central dogma works. Now, fibroblast growth factor receptor 3 is expressed in resting chondrocytes that make, and they inhibit cell proliferation and differentiation. Their overactivation then will inhibit chondrocyte growth, and so you get short limbs. Greater activation, the shorter the limbs, and other more severe skeletal dysplasias. So, potential therapies, you block the activity, but not completely. Then you get other things like craniosynostosis syndromes. You get premature fusion of cranial sutures, leads to misshapen skulls and limb defects. Again, another autosomal dominant mutation in one of these three receptors. The same mutation can cause two different syndromes, and then the phenotype is going to be affected by a modifier gene. So when you're reading some of these things, you have to take into account, is it activating, is it inhibiting, in order to really understand what it's doing. And this is what I'm trying to do. I'm not only trying to give you an understanding of how the DNA genes are in the DNA and how that works, but then be able to relate that to the actual protein, its function in the cell, and then its disruption of its function that leads to disease in your patient. And so this is just some of the, another example in fibroblast growth factor, and you can see a child with Apert syndrome. You can look at these. I'm not going to test you on them, but I just want to give you a sense of how miraculous and wonderful this topic is. I just wish we could spend like the whole thing on it, but of course then we can get all the other cool stuff as well. So we're going to talk briefly about pattern formation. It's defined as the process by which ordered spatial arrangements of differentiated cells create tissues and organs. General pattern of body is laid down during embryogenesis, and these lead to different regions in the embryo, or semi-autonomous. Oh, I swear, I'm getting the emphasis on the wrong syllable a lot in this one. Anyway, there's also regional specification, definition of the cells within a region. What cells compose a region then? Then you have to have signaling centers. These give positional information, and then they can differentiate, of course, again in response to cues. So it's always going to be, with communication, a signal, a receptor, signal transduction, change in gene expression. So this is why the central dogma is so essential to understanding all this. So communication is critical for pattern formation, and of course it's a very, very active area of research, because if we can learn how to do that, not just only in embryo but in adult stem cells, think of what we could do to repair spinal cords and like that. So I have to tell you about the hedgehog family. They've got a little bit in the textbook here as well. But this was identified from a Drosophila mutant. Drosophila is fruit fly. Okay. And it was, as I said earlier, it was supposed to have bristles and be smooth, but it has bristles. So the guy must have been British, so he said it looks like a hedgehog. Anyway, in vertebrates, we have something called sonic hedgehog, or SHH. And it's very important in specifying the axis. It's important in induction of the motor neuron, okay, and patterning limbs. So we're also going to now what it does in adult humans, it's called, or developing, is it binds to the sonic hedgehog SHH receptor. SHH is a signal then. SHH receptor is called patched. And it inhibits a transmembrane protein called smoothened. Now, if SHH binds to cholesterol, it binds to patch, releases smoothened, okay, which activates a signal transduction pathway. If this doesn't happen, okay, then things will not get developed properly. And these are some real critical areas of development, patterning of limbs and motor neurons. And so some of the disorders that you're going to see in the next slide are really very upsetting. Now, you can also have a mutation which may lead to cancer because it's removing an inhibitor. Okay, if patch is always on, regardless of whether sonic hedgehog SHH is binding, then the cell is always being activated, and it could achieve other mutations that lead to a cancer phenotype. So these are some of the pictures with the problems with sonic hedgehog and patched, but the next page has tells you more about the neural tube here, and of course its effect that way, and you can get this holoprosencephaly, severely mentally retarded, very early deaths, but there are milder forms as well. And again, it requires attachment to cholesterol, and it just shows you some others as well. So I'm pulling in some slides from some other places just to give you a sense of how important this signaling is. Wingless is a Drosophila mutant. There's a vertebrate homolog, we call it integrated, establishes polarity during limb formation in flies and a similar function in us. And again, there are secreted glycoproteins that are involved. They bind to a receptor, again, LDL receptor related proteins. 19 of these different receptors and stuff are in humans, and they have a wide variety of developmental processes within humans. Again, specification dorsal ventral, formation of brain, muscle, kidneys, gonads, and there are homozygous mutations in one of them, wingless 3, which causes tetraamelia, absence of all 4 limbs. And so I couldn't resist putting up this particular picture of Nick Vujicic. I think that's how you pronounce his name. He's a motivational speaker. He has this defect, and if you've ever heard him speak, go to YouTube, and he is amazing. DNA transcription factors are also important. They turn on and off transcription of several genes, and this is ultimately where it's at. By turning the genes on and off at the right time, the cells go to the right spot, they differentiate into the right type of cells, everything. And of course, turning on transcription is turning on and off synthesis of messenger RNA and ribosomal RNA so we can make the proteins. They frequently function in a cascade, so mutations in DNA transcription factors, because they can bind to many different promoters and many different genes, will have pleiotropic effects, lots of effects. And of course, they all have to be able to bind to DNA. So they all have a portion of the protein that binds to DNA, which is known as a DNA binding protein, or a DNA binding domain. Anyway, the roles of these are controlling development, and mutation cause some real serious birth defects. There are, of course, going to be more than one type of transcription factor family that is involved in development. So you've got the homeobox containing genes. These are the HOX, PAX. High mobility, these are called HMGs. or high mobility box containing genes, sometimes SOX, and another family called T box. And these are important for getting these patterning early on in the embryo and later on as well. And particularly you can see here with whether they're developing the spinal cord and the brain and everything. Extracellular matrix proteins are just as important. These are secreted macromolecules that serve as scaffolding for all tissues and organs. They're going to be things like collagens, fibrillins, and we've seen that in Marfan syndrome. Proteoglycans, large glycoproteins, such as fibronectin, laminin, and tenacin. So these are things that some of you have picked up in terms of different types of disorders, and why I kind of want to bring them in to help you with your summary portion paragraph of your discussion. So they're active mediators of development. They separate adjacent groups of cells. They form matrices in which the cells can migrate, so it's kind of a platform. Fibrillin 1 and elastin coordinate microfibril assembly within the extracellular matrix, so that's very important. And so, as I've already said, Marfan syndrome with mutations in fibrillin 1. You also have supravalvular aortic stenosis when you have mutations in elastin, and both can also lead to other congenital heart abnormalities. And so I'm just going to very briefly go through some of these major events during embryogenesis. So gastrulation, this is the process of cell and tissue movements where the cells of the blastae are rearranged so they have new positions and new neighbors. It's day 14 to 28 in humans. And so you get the three germ layers that I think most of us have heard: ectoderm, mesoderm, and endoderm. And then this forms a primitive streak, so we get anterior and posterior axis. And then we start beginning to form the extraembryonic tissues. And many of the genes expressed then, not surprisingly, are genes that will be expressed in adults with cells that move, like the white blood cells, etc. So this is just a picture of it. You can take a look at it in a little bit more detail, but I'm not going to go into it, and I'm certainly not going to test it. I just want you to appreciate and marvel at what these genes are doing. So, neurulation, now we're getting the formation of the hollow neural tube. Of course, we need induction, organogenesis to do this, and this is going to divide the ectoderm into 3 different cell populations. The neural tube, which of course is critical for the brain and spinal cord. Then we also have the epidermis and those neural crest cells. In humans, the neural tube should close at 5, the beginning of it closing should be at 5 different sites, and of course these different sites correspond to common neural tube defects. So you can have anencephaly, absolutely no brain, occipital encephalocele, then you have a herniation of the meninges or brain, and of course lumbar spina bifida. The neural crest cells will migrate and differentiate into the sensory neurons and melanocytes and neurons of the small bowel and smooth muscle. So these are very important as well. And you again begin to see the patterning of defects in a gene that is important or derives from the neural crest with the symptoms that the patient exhibit. So mesoderm and endoderm. Mesoderm's in 5 components. You've got all these others, and you can see what I'm doing is just showing you whenever you read about this stuff or are looking it up perhaps in your disorder. This is a resource for you, so I'm not going to really go into it in detail here, but this allows you to easily look at this to see if you're trying to figure out something with your disorder. The endoderm, of course, the digestive respiratory tract, and budding and branchials. And again, here's the fibroblast growth factors that we're talking about. Axis specification, of course, is very important. This is going to give us left and right, anterior, posterior, and dorsal ventral. And say the proteins mediating the axes are rapidly being discovered. So we're still investigating a lot of these. but they also have additional roles in patterning the body plan and tissues. And here are anterior posterior axes are determined by some of those Hox genes. And dorsal ventral axis, again, those signals. And then you also have the organogenesis. And this gives you just a little picture down here of the left-right symmetry and how it can get mixed up. We've all heard about the people who, they're supposed to have everything right and they are completely reversed. It's in these homeobox or homeotic genes that you read about in this chapter. Craniofacial developments directly related to the formation of the central nervous system. So the neural crest cells form the forebrain, midbrain, and you can then read how that works. So all determined again by the Hox genes. So now you see why facial features are so important in some of this. Bones of the skull develop from the neural crest as well, and you can have premature fusion, so you get misshapen heads, and of course that can impair brain growth. Here you go, fibroblast growth factor receptor. Development of the limbs. This is the best understood. It's the classical model of development. We've isolated and characterized many of them. Obviously, some of them are going to be involved in the signaling pathways and transcription. And we found many of them in Drosophila, so we're also seeing them in humans as well. And incidence of newborn limb defects second to congenital heart defects. That's how common it is. And this is showing you the ulnar mammary syndrome. And then organ formation. So again, cellular interactions and communications. Again, you need secreted signaling molecules binding to receptors, inducing signal transduction, changing gene expression by activating or inhibiting function of other transcription factors. Once the organ's formed, you frequently have signaling molecules to induce the cells to function. Bind to receptors again. In this case, it can be on or in the cell. What's it going to do? It's going to change gene function. And this is the thing that goes on and on. So you can see why I get real excited. I hope you can get a feel for how exciting and fascinating this particular topic is. And we're beginning to understand it more and more. And for someone like me, that means it's more and more miraculous. Because when we finally figure something out, it usually makes a lot more sense than when we don't know it, because we don't really understand what's going on. But then when we do, we can see the beauty of the complexity, and in many cases, the simplicity of it as well. All right, well, I'm going to leave you with this particular topic at this point. You'll get to hear my lovely voice again. I always feel sorry for you guys at this point in Chapter 13. Take care.